So, Ms. Mathew, thank you so much for taking out the time and speaking to our students. Students, piece of advice, please take this opportunity and gauge the maximum and ask as many questions as possible. Thank you so much, ma'am. All yours. Thank you.
Thank you, Shweta. It's my pleasure. Just too many kind words, but it's my pleasure to be here to address. So, how many people have around joined? Around 116, I
116
can say.
and still joining.
Yeah. So, I've made a very brief deck of slides on regulatory just to give understanding on. I know some of you have a background on regulatory. A couple of slides on people who want to take up regulatory as their career. And so, I always, wherever I go, I always say regulatory is the most dry topic to be taken virtually. So, people sleep off people because otherwise, it's very difficult to understand. Like, people are grasping it or not. So I've made it very simple for people to, gauge through what I'm talking. Please stop me and ask in between, because at the end of the session, I know we have a Q&A session, but it doesn't, take very effective. So just stop me in between. Again, you speak if you have any doubts on the slides. To give you a very, not my intro, but then how I joined regulatory and how my regulatory journey started. My basic background, I started after my master's was in R&D and in biomarine in UK. I did my master's in London, and then from there I was with them for 2.5 years. So regulatory has been a very close to heart department because we had a very strong technically driven people there. And for me to get into regulatory was very impossible at that time because you required experience. So since then, this has always been back of my head that I didn't want to continue in R&D and I wanted to shift to regulatory, because regulatory, I understood, because regulatory is one domain which cross functions across all the domains of a pharmaceutical industry, which will come in the subsequent slides. And that is how regulatory happened for me. I would love to share my experience and how, so I don't want it to be a only technical talk. So please pitch in and ask questions. So this is my agenda for today, to give you a quick intro into the pharma sector paradigm, introduction to regulatory aspects, RA from a career perspectives, the differences between the general terminologies about modules, generics, and biosimilars, overview of the regulatory framework, summary, and conclusion. So to start with, any pharmaceutical, so right, there are multiple pharmaceutical industries in India, in abroad, all the other countries. We have MNCs, we have domestic markets. So a lot of companies which are manufacturing and commercializing drugs. So the entire, the vision that binds the pharmaceutical industries, the companies with a vision, it's innovation, competition, options, and access. So the overall, the insight is to provide more treatment options for patients, greater access to life-saving drugs, potential increase in market competition. When I say market competition, it's again, stems up from the fact that you have a novel molecule in the market. Novel means anything new drug which does not exist in the market is called a novel molecule, right? So you put across a novel molecule, the marketing exclusivity of the novel molecule finishes, and then comes the generics, the biosimilars, all into the market. So then the agencies, the governments of the countries, they basically promote more and more companies to file as generic versions or as biosimilar versions of the existing drug in the markets, thereby increasing the market competition and access of the drugs. The global pharmaceutical market is, just to give you a very brief understanding of how huge is the pharmaceutical industry. The global pharmaceutical market is estimated at an astronomical US dollar 1 trillion in value currently. The key driving factors obviously are the research, development, and then innovative treatments for patients. Now, most of you would be aware of something called the AI, the artificial intelligence that has been driving and has been a hot topic of the recent times. So, whenever, we discuss, people ask questions, is AI going to displace, the human workforce? It is not going to displace human workforce, but what I would say is that in the coming times, it would be always preferable for candidates who are AI equipped. So I would say, from the company that I'm working on currently, so making everything into AI enabled. So probably another 5 years target, we would have dossiers being reviewed by AI tools. We will have everything. So even for the with the basic things like you might be aware. So every time we have this meeting and we write the minutes of meeting. So we don't write the minutes of meeting now. So it's AI enabled, right? So there is something called Copilot, and that gives you a summarizes the entire minutes of meeting. Very basic example. So AI and big data technologies will they're basically allowing us to restructure the entire business from a perspective of efficacy, time management, and human resource management. So 10 tasks that we are doing, the time that you take for 10 tasks would be 10 hours. So that gets considerably reduced when you use an AI enabled platforms. So that is something of very big interest and very big scope that is coming. Not exclusive doing AI courses or anything like that. That would be always an add-on, but understanding the AI dynamics, how you can implement AI in your current workforce is a very important factor that would be upcoming in the coming years. India has already built a very stable foundation for the local and global growth in terms of the indigenous growth pharma sector. When I say we have indigenous means native, right? So we have a lot of companies which are producing drugs earlier which we did not have access to in our domestic market itself. Right. This slide is for just to give you a quick, very fine line difference between the regulatory affairs and regulatory science. So generally in Indian companies, you would not find a difference in this, but there are some companies abroad and some very few companies where there is a difference in the discipline between regulatory affairs and regulatory science. Okay, so the regulatory affairs is developed to protect public health by controlling the safety and efficacy of the products. The regulatory professional should have a mix of scientific knowledge, negotiation skills, business intelligence, which is very important part of being a regulatory affairs candidate. Now, when it comes to regulatory science, this is basically a mix of range of the scientific disciplines that are applied to the quality, efficacy, and safety. Now, to understand the difference, there's a way, there are people who come back with background for medicinal chemistry, who come up to be as medical writers, who come up as clinical reviewers. So all these falls under RA, falls under the regulatory science. So this basically are people with basic and applied medical science and contributes to the development of regulatory standards and tools. So if at all you come across these both terminologies, this is the very fine line difference between both. Let me know if I'm fast, if I need to tone down, because I speak a little fast, so just hold me in between if I am. So this is a slide on pharmaceutical cross function chains. So when I was initially in R&D department, so I was basically a cell and molecular biologist. When I shifted to regulatory, even then when I was in R&D, my understanding about the dynamics of the pharmaceutical industry was very limited. I knew QA existed, I knew QC existed, production existed, and R&D existed, and marketing existed, obviously. So these were the major domains in my understanding, which were part of the pharmaceutical industry. But when I joined regulatory back in India, I joined Biocon. That is when I understood there are so many subsets in the pharmaceutical cross-function chain which contributes to an approval of a drug, to the development and approval of a drug and commercializing of the drug. For example, I have only quoted few of the verticals. I have put global regulatory affairs in the between. I will tell you why. So we have quality assurance, we have quality control. So quality control basically checks the results, the data. Quality assurance basically assures that the data is correct. So then you have R&D, wherein they are developing the drugs, wherein they innovate, science driven department. Then we have product development. Then we have something called supply chain, which manages the entire drug dispatches, the forecasting, the market forecasting, everything. Then we have something called clinical. So we have non-clinical, the clinical department, we have manufacturing, we have commercial. So now there is also something called IPQC, in-process quality control. We have in-process quality assurance. We have microbiology, we see QCMB, quality control microbiology. So all these are very, very busy departments which contribute their big hand in the developing of the, in the assistance in the developing of the drugs into the, final formulated stage. So it's a very interesting aspect to understand the entire cross-function chain. Now, why have I put the regulatory affairs being the central brick of all this? I'll tell you why. So regulatory is one department which has to integrate with all these departments, because R&D develops the drug, manufacturing produces the drug, supply chain, does its own, then the commercial part. So who files the drug? The global regulatory affairs files the drug and gets the approval, right? So when we file the drug, we file for the approval. All this technical data that we have from all these cross-function departments come to us. We review the data. Not that you are technically required to review an analytical technology, but it's always an add-on if you can do that. But at the end, from a regulatory, suppose for example US FDA requirement perspective, are we giving the data required for the typical agency? It can be US FDA, it can be Europe, wherever, whichever country we are planning to file, we have a list of requirements from the country for the approval. So from aspect of that, are we meeting the requirements? Are we giving the required set of data, required set of standards that the agency requires? Is what we do. What we basically do is we get on all the data from this all multidisciplinary cross functions, make it into something called a dossier. We call the dossier. Dossier is basically nothing, a set of documents that we submit to the agency. Now you have your drug substance, we have drug product, how we formulate the drug substance into drug product and in a consumable form. How do we do that? So this entire, it's like a stepwise approach to convince the agency that we have an safe and effective drug in market with no side effects, with no adverse events. So that is what we do. So this is basically the entire pharmaceutical cross function chain looks like. And when people, I don't know whether you're freshers or people have started working, so when you get into the corporate space, you basically have an understanding of how the interacts with all these cross function teams to get into the approval space. So now regulatory affairs and its role. And so this slide is basically just to give you a quick understanding about how what was the need of evolution of the regulatory affairs. How did it happen? So we basically are a profession which acts as an interface between the pharmaceutical industry and the drug regulatory authorities across the world. So it's not like it's so teams are split. In certain companies they have different disciplines. So in the company that I'm working currently, we handle global, so it's like one product in 90 plus countries I handle. So but in Biocon it was different. So we had something called regulated markets, we had India market was different. So it depends on the company on how they practice their regulatory affairs team. So I don't know whether you guys are aware, must be aware of the biggest, disaster that had happened in the world called developed by the thalidomide. This was the biggest man-made medical disaster. What thalidomide was, referred for pneumonia, viral fevers, and all these kind of fevers. So a lot of deaths happened, and almost 10,000 kids were born with abnormal fetal abnormalities. So that is when, in October 2, in US, the Congress unanimously passed the Q4 Harris Amendment, which basically addressed the shortcomings in the regulatory policies and regulatory laws that we had in place, because till then there were a lot of shortfalls. So how is it, for example, I'll tell you about US FDA. So when a typical pharmaceutical company used to apply for a drug approval to US FDA, within 60 days, if the company does not hear from US FDA, they will start manufacturing and commercializing the drug. Once the drug is in the market, then you don't have a control over it, right? So this was how, there were a lot of loopholes in the regulatory structure then. So risen with this amendment, the other changes that became the solid foundation for the regulatory approvals for across the global was it required that manufacturers prove the efficacy of drugs before they go to the market, requires that this evidence be based on adequate, well-controlled clinical studies conducted by qualified experts, requires informed consents from study participants. When I'm saying the study participants is the clinical study participants, people who give, who dedicate themselves for the being the subject for a clinical study. gives FDA 180 days to approve or reject a new drug application, allows the FDA to set good manufacturing practices, which is a GMP. So now if you want to manufacture a drug, that facility has to be GMP approved, right? So all these laws came into place because of some tragedy and thousands of deaths that happened, and then they formulated the entire regulatory laws, and and it started reflecting across globally. So now the slide is quickly. Any questions so far? If anyone has, please ask before I get into the next slide. So let me tell you, right? Um, Questions does not have to be brainy questions. It can be silly questions, because if you don't address the silly questions, you will never get, the understanding of the holistic picture of the entire thing. It's a very simple thing, but ask questions. So that is why even I will be, it'll be a conversation-driven presentation. So drug development is a stepwise approach. Why do we say? So right from the innovation that happens in R&D, then we characterize the drug, we develop analytical methods, we make it into, then the API, the active pharmaceutical ingredient, which is the drug substance, is formed. Then comes your DP, which is the drug product, which we make it into a consumable form for the patients, and the people in the world. So this is basically a stepwise evidence approach. It's not like you start something and then you have a drug. It's basically very iterative process of finally before you have your drug. Know your product, keeping it in mind. So this is something I always think, because my professor used to keep telling, who he used to take classes for us in medicinal chemistry. He used to tell, anytime you narrow down on the company narrows down on developing a drug against, either it can be a novel drug, or either it can be a copy of a existing drug, you should always have the end in mind against which you will prepare your trajectory to develop a drug. So this is like a very common mantra that we use in the pharma industry: know your product, keeping up in mind end in mind. You might be aware also of this ICH guidances. This ICH guidances are basically the Bible of regulators. So we follow upon all the ICH guidances. So for anything, for safety, for efficacy, for the chemistry, manufacturing, any aspect of your drug development, we always refer the ICH guidances. ICH stands for International Council for Harmonization of Technical Requirements for Pharmaceutical for Human Use. It was founded officially in 1990. Basically, the entire concept is to promote public health through international harmonization. So just like it's very important for you to have standards, right? So every country has its own regulatory. So suppose India has its own regulatory requirements. China has its own regulatory requirements. EMA has its own, EMA as in European Medicine Agency. US FDA has its own. So when each country practices its own regulatory requirements, ICH is one umbrella which basically controls, which basically not controls, for harmonizes the regulatory requirements across the global regions. So this is basically for developing and manufacturing of new drugs, registration and supervision of new drugs, prevention of unnecessary duplicate. I'll come into the details of this in the subsequent slides. So all these to harmonize the regulatory requirements. That is why the ICH, as an umbrella, it's in picture. Okay. So now, this is a slide I wanted to quickly give you an understanding between biological and small molecule drugs. So there are three terminologies where you generally come across when you go to company, when you work in companies. You have novel molecules. So there are companies like Novo Nordisk, where I'm working. They are only specializing in new medicines, novel molecules. The novel molecules is a molecule which does not have a similar molecule in the market. It's a new molecule, right? So we invent, we innovate, we invent, we market it across as a new product in the market. Now, what does Biocon do? Companies like Biocon, companies like Intas, companies like Dr. Reddy's, all these companies are the generic companies, right? So they make identical copies, sorry, they make the identical copies of the already existing drug. Okay, so now suppose a drug A is there in the market for, all right, let me take an example of Herceptin. Herceptin was a drug for developed by Roche, which was a drug for breast cancer. Okay, so now in 2014, 16, I suppose, Biocon developed the biosimilar version of this existing drug of Herceptin, which was called trastuzumab. I don't know, it was all over the paper and all over the news. You must have read over it. So trastuzumab was a blockbuster drug, biosimilar. It's called a biosimilar. So now you have novel, you have something called small molecule generic, you have something called biosimilar. Now the difference between generic, small molecule, and biosimilar, paracetamol, for example, aspirin, all these are chemically produced drugs, right? Small molecules are basically drugs which are chemically produced. So you can see the differences. Low molecular weight complexity, why is it is very low? It is chemically synthesized. The manufacturing process is easy to reverse engineer. When I say, so I don't know how many people of you know the entire, the drug product formulation. So you have DS, we add excipients to it because DS cannot be, drug substance cannot be consumed in its own form, right? So we add excipients to the drug substance and your drug product is formed, right? So when it's a chemically synthesized small molecule drug, for example, aspirin, okay? So they are very low molecular weight of, and generally very less complex in nature. So a plus B gives you C. Now you can break down C back to A plus B. That is why we say manufacturing process is very easy to reverse engineer. Now, this one we call it as generic apple to apple molecule. So now suppose you are developing a generic version of an aspirin, you will develop an exact molecule of the aspirin that is there on the market because you know what is the constituents and you can just add the chemicals and excipients and then you have aspirin. But when it comes to biosimilar molecules, biological molecules are very complex in nature because of their route of synthesis. Now, biological molecules are basically molecules which are produced by recombinant DNA technology, right? So, recombinant DNA technology, I don't know how many of you are aware, in that clones are introduced, in which we clone a typical gene and then we allow it to, we insert that into a required, organism like E. coli or Pasteuria, Pichia pasteurus, we introduce in it and we allow it to flourish in a fermenter, and then we extract the protein out and then reformulate and make it into a consumable form. This is the entire process. Now, why is it complex? His reason is because clones are involved. No two living cells can be similar. It has its own inherent variability within. The reason why it is called biosimilar and not bioidentical. Now, generic small molecules, we call it as apple to apple identical product to the novel. Biosimilar, we call it as biosimilar because it cannot be identical because it is originating through a recombinant DNA technology and it has inherent variability. Because of that, it is called biosimilar. Now, how do you prove whether the biosimilar drug that you're developing is equivalent to the novel product? Yes, definitely you have to prove that your product is equivalent to the existing novel product. For example, trastuzumab was a biological medicine which was developed for breast cancer as a biosimilar to the existing Herceptin in market. Now Biocon had to prove that. So we do a multifold testing and analytical comparability, lot of testing to compare the existing R framework with the existing. So for that, there's a lot of work in the R&D that happens for a biological molecule. If a generic small molecule takes 3 to 4 years to develop, a biosimilar takes at least around 8 to 10 years to develop and come into the market. That is the difference, and there is a lot of investment also involved because it's more expensive molecule. So now, um, to give you a quick idea on the figure of the molecules, very very rough idea, not the exact probably. When Herceptin was introduced as a novel molecule for the breast cancer in the market, you know how much was the price of a Herceptin? It's a single 400 mg vial, single dosage, which was given to the breast cancer patients. It was roughly around, might not be the apt figure, but just to give you an idea, it was 98,000 Indian rupees for a single dosage vial. Now, when the biosimilar came into picture, so Vicon thought at that time 400 mg was not required. So we made a 120 mg vial, wherein they costed around 28,000. So this is the price clash. between the novel molecule and the biosimilar molecule when it's in the market. Still, it is out of, how much of percentage of the Indian population can afford a 28,000 single, dosage. That is a different chapter altogether. But what I want you to understand is the reason why health authorities and governments promote more and more generic companies and biosimilar companies to come into picture and to produce drugs is also for the affordability. So now from 98,000 Indian rupees to 28,000 Indian rupees, it's a big margin cutoff, right? So this is what happens when you get a biosimilar in the market, when you get a small molecule generic in the market. So now the bottom diagram is just to give you a quick understanding of what I explained before. How does the biological molecule gets produced? We cloning into the DNA vector, mammalian cell produces protein, then we have fermentation, we extract the protein out, we do the formulation, and then in the consumable form, in the form of injectables or however it is, we formulate it. Okay, so this is to give you a quick understanding on the differences between large molecule drug and the small molecule drug. Any questions on the slide, please?
Ma'am, Soumya here.
Yes.
Ma'am, you said the biosimilar production made a huge difference in the price because of the vial. They reduced the quantity of the vial.
No, 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 not because of that. That was one specific case. What I meant was, even if it is any biosimilar that comes into the picture, right? They have the novel product that is in the market is very expensive, because the reason why I will tell you, they have to run extensive clinical phase 1, phase 2, phase 3. They have to run the entire clinical studies, non-clinical studies, because they don't have a reference product, right? They're making a new product. So all that cost taken into effective, they marginalize their product and put it across in the market. Now, when you're finding a biosimilar or a generic in market, you do not have to, as a biosimilar, you do not have to do the extensive clinical studies like the innovator. Because if you open, I don't know, you can always check. If you have not checked, you can open the US FDA website, you can give this Herceptin, for example. Herceptin, if you give, you can get the entire innovator data. Innovator, what they have done. Probably, some of the things will be masked, as in it's because of the confidentiality thing. But we get the data. So we rely on the innovator's clinical study, and we only do a very minimum bridging study to bridge, to understand, our molecule is similar to the novel molecule. From that aspect, there is a lot of cost difference for us than a novel molecule. That is why a biosimilar is always less expensive than a novel molecule.
Yes, ma'am. Actually, I got it. My question was why can't the novel production company, why can't they give it in the different ml sizes? Or
that was their strategy. This was not a needed information which I told you. This was Biocon's strategy for getting into for their market purposes. So innovator can do that. Innovator can always do that in whatever sizes. But the context of my conversation was the pricing differences between the biosimilar and the novel. The reason why.
okay, so since we don't need extensive clinical trials for biosimilar, that also helps in the product pricing of biosimilars. That's it.
That also helps in the cost cutting for biosimilars.
Thank you.
Yeah.
Thank you.
Any other questions?
No, that's it from my side.
Hello,
Yeah.
Nikita
Nikita.
Govnikal. Yes, ma'am. Because of the cost, we can make the biosimilar and the generic version of the drug so that anyone can afford this. This is the main agenda for making this
That's
biosimilar
why
and
the health authorities and the companies generally promote the more of biosimilars and more of generics, so that it becomes more affordable for the patients.
okay, means affordable. The
Yes,
main target is
affordability from affordable and accessible, to the community.
okay, ma'am. Got it, ma'am.
Yeah.
Thank you so much.
Ma'am,
Anyone else? Anything?
excuse me, ma'am.
Mm-hmm.
Good afternoon. Myself Disha Gupta. Ma'am, will the effects of the drug will they not vary? The effects of the
I,
drug on the
I
novel
I
drug,
don't
the
think I hear you
biosimilar?
very well. Your voice is breaking. Shweta, can you help me understand the question? What is it?
Sure, sure. Disha, can you just speak aram se? Your voice is muffled and echoing.
Okay, sorry.
No, no,
Can you
don't be
hear
sorry.
me now?
Yes.
Yes,
And
ma'am.
also
So
type your question.
my question was the effects of the drug, of the biosimilar novel drug, will they vary also?
The effects of the drug with respect
Like,
to the novel, does it vary? Is it your question?
yes. Yes,
Okay.
ma'am. Yes.
So that is why agencies have FDA, US FDA, EMA, and all the country agencies. They have guidelines. Okay, so they have biosimilar guidances wherein we have to give studies, reports, data. The primary data that we give is the analytical studies. Okay, so now you have a biological structure. You see this thing, the large biological molecule structure over here, the immunoglobulin. So this now immunoglobulin getting developed. You have primary structure, you have secondary structure, you have tertiary, you have quaternary structure. Those who are related to the biological molecule will make a sense of it. So now, right from the start, we have to study the primary structure of the novel molecule. So what we do is first we buy the novel molecule from the market and we study it. Okay, we characterize the molecule, and then we have our molecule, in-house molecule, this molecule and this molecule we compare. Unless and until you have a similar molecule in terms of efficacy, you will not get approval. Okay, so we have analytical studies that we give bridging clinical studies, we give immunogenicity studies to prove that our drug does not have a different immunogenic response than the existing drug. It cannot be identical in terms of the drug properties, but it has to be similar in terms of the efficacy and there is no, because otherwise the patients would get adverse events and all those issues will come, and then you will have to recall your product. So an agency never gives approval to the product unless and until you prove compatibility of your drug to the existing novel product. Go to the next slide, or anyone else here has any questions?
Yes, Kankara here. So I have one question, if I'm not wrong. So biosimilar doesn't require clinical trial, is it?
No, it requires clinical studies, but not as extensive phase one, phase two, and phase three. So generally, it requires a phase one, and some of the molecules it requires a phase three study.
That is.
The insulin
And
molecules, the insulin molecules which has been in the market, right? So now for the insulin has been a legacy molecule, right? Since almost like 100 years, it's there in the market now. So now for insulins, Europe Medicine Agency has waived off the immunogenicity study till that year, till I think 3 years, 2018, I think till 2018 you had to do the immunogenicity clinical study also. But now, because insulin is a legacy product and they know insulin structure and insulin, the general requirements that you give, you don't need to do. But there are certain bridging clinical study that you need to do mandatorily for all biological medicines.
okay, so post marketing surveillance.
Post-marketing surveillance is across the molecules, whether it's a novel, whether it's a
Mm,
biosimilar molecule, right?
okay, so it will
Post,
happen for the biosimilar also.
sorry?
It will happen for the biosimilars also.
Yes, yes, yes. So
Yeah.
post-marketing surveillance is how much ever, see your phase one, phase two, and phase three subjects. Okay, so now for phase one you might have 700 subjects. Your phase three maximum you'll have is 1,000 or 2,000, depending on the molecule. That number keeps changing. But now this is a subset of samples or subjects on which you're studying your drug. Now the concept of pharmacovigilance is when it's outside in the market, that is when the mass clinical study happens. That's called the phase four, right? So now when the patients consume it, you see the toll-free numbers on your medicines, right? Which we
Mm,
Indians fail to understand, fail to see even sometimes. Every medicine has a toll-free number. The toll-free number is a manufacturer's number. Okay, so now for example, DRL is producing a drug and is in the market. Now you have a toll-free number. Why is that toll-free number for? The reason is, suppose you're consuming a biosimilar product or whatever medicine it is, you get an adverse event as in the patient information leaflet that is there in the product, right? So in the
mm,
patient information leaflet we have a list of contraindications, like you might experience nausea, you might experience loose motions, diarrhea, blah blah blah. So apart from that, any of the adverse events that the patient suffers, now you have to call up the toll free number and notify the manufacturer telling that the patient A is suffering from adverse event. The manufacturer is bound by law to notify US FDA within 15 days that adverse event 1 has picked up. Okay. Now, similar like this adverse event gets, that is when your product gets recalled. Okay. There are chances where immunogenicity gets a concern for some of the drugs or some of the adverse event which certain clinical studies would not have captured. When it's out in the mass public, that is when you understand. There are a lot of cases. If you will want to read, Johnson's had a lot of product recall cases. You can just Google and read it. That is the reason you have pharmacovigilance across the products, irrespective of the nature of the product.
Okay, ma'am. Got it.
That answers your question?
Thank you.
I'm happy I heard some questions finally. So now, as I was telling earlier, the standards that the generic or the biosimilar medicines needs to meet to receive approval. These are the same. These are the regular standards. Okay. Apart from that, there are a lot of regular data, but these are the regular standards that you need to meet for you even to qualify to prove the sameness of your, to the novel drug. API, which is active pharmaceutical ingredient, or we call it as the drug substance, or the active moiety, should be same as that of the innovator drug. Innovator, the different usages of innovator, it can be called as a novel molecule, it can be called as a reference listed drug, it can be called as a reference medicinal product, which is a common usage in Europe. It has to have the same strength. If it is 6 mg per ml, the innovator, you can only develop 6 mg per ml. You have same route of administration. When it's an oral drug, if the innovator is an oral drug, you can only develop an oral drug. If it's an injectable, you have to develop an injectable. Same inactive ingredients. Inactive, when I say, you have active pharmaceutical ingredient, which is your active moiety. Along with that, the excipients that you add along, those are called the inactive ingredients. Suppose you have 5 inactive ingredients like hydrochloric acid, the buffers, the tonicity agents, all these that you add, everything has to be in equal qualitative and quantitative composition equal to that of the novel. Now, where do we get this data? If you wonder, everything is there in the US FDA website, the Europe Medicine Agency, for the novel drug, everything is there. So against which we develop our drugs. Same dosage form, same label as the brand name medicines label. Label as I told you earlier, the patient information leaflet that is there in the medicine has to be exactly same as that of the innovator. Meets the same batch requirements for identity, strength, purity, and quality. Now, batch requirements, when I say, when we manufacture, in batches we manufacture, right, the drug product, the drug substance, so we give data of all these batches to the agency for approval. Now, one batch, for example, the pH value we maintain between 3 to 4. One batch is having pH 3 to 4, another batch is coming 5 to 6, another batch is coming 6 to 7. It will be rejected. You cannot have a batch consistency like that. The pH has to be consistent across the batches that you're manufacturing. Okay, so this is to give you a quick overview on the difference between the novel and the biosimilar or the generic versions that we submit. So now, standalone development program, when I say it is a novel product that we are filing with US FDA, the application form is called 351A. Now you see the green box, okay? So the thick part is the top portion is the clinical safety efficacy, phase 3, then we have phase 1, phase 2, then we have animal analytical. So this is like the chunk of data that you have to give for a novel product. Now, when you come to abbreviated, why do we call it as abbreviated? Because it is not an entire exhaustive data, but we are making abbreviated, a shorter form of the existing drug, drug dossier, right? So that application form is called 351K, not for you to remember, but just for you to give you an overview of how US FDA takes through this, applications. So now, if when you're filing a biosimilar, it has to go through a 351K application. Now, what is your goal? Goal is to demonstrate biosimilarity to an existing reference product. Reference product is nothing but your novel or your innovator product. Now, look at this is like a triangle, right? Do you know why? Because over here, your analytical data, as I said to you earlier, the primary structure, the secondary structure, the tertiary, the quaternary structure, your data masses, every analytical study that forms your base of your dossier. And that happens only when you characterize the novel molecule and we characterize our molecule and we understand this is the character. So this is basically a comparability data that you give with respect to the novel molecule. Okay. So then comes the animal data. Now, there's something that I wanted to give you a quick information on that. I have 10 minutes to wind up. I'll quickly wind it up. So, on animal data is also called preclinical study, preclinical data. It's also called toxicology data. It's called non-clinical study, different terminologies. Why do we do animal data? Animal data, we basically call it as a proof of concept study, right? Because when a novel is in market, when it's a new drug, you test it on the animals to understand the lethal toxicity, the lethal dosage level, everything, right? Suppose 50 mg, very layman example, I'm just giving you 50 mg, we are giving it to an animal, the animal shows stress. Then we know that it is toxic to the animal. So we reduce the dosage. So basically, to understand the toxicity levels of, so you cannot do it directly on humans. So we leverage the data from an animal study and we leverage it to the human study. Okay, so now all these data is already existing for a novel molecule. For generally for biosimilar or a generic molecule, we do not do animal data. And now USFDA, a lot of agencies are coming up with a something. There is something called the FDA Modernization Act. Read it. It's quite interesting, and just Google it. FDA Modernization Act. According to that, what they're saying is they're trying to work on ways to reduce or remove the animal studies, basically, wherein they use softwares like in silico modeling, statistical data packages, which will mimic the already existing. We have a lot of consortium of animal data, right? So against the typical indication, basically a statistical driven package to understand how the molecule is. So that you don't use this is basically arising from the animal cruelty causes and all that. So generally we don't give animal data for biosimilar. So then we have clinical study as the bridging study and additional clinical study if the agency asks you to do. So this is a quick snapshot between difference between a standalone and abbreviated. Let's skip this study. So this is also, I'll share the slides with Shweta. Just go through it. If anybody has a doubt, just reach out to me. So this is basically just to show you why do biotechnological products, the biological products, has inherent challenges. As I told you earlier, it has multiple levels to structure, multiple active sites, post-transition modifications, susceptible to multiple degradable pathways. That is the reason the agencies are very, very typical and very strict when it comes to a biological molecule approval than a generic simple molecule approval. Animal data, I have explained to you. Role of clinical studies, again, it depends on what do we expect from a novel molecule and what does the agency expect from a biosimilar molecule. FDA expects an adequate clinical PK, pharmacokinetic and pharmacodynamic. It again, so there is no standalone, there are guidances which agency has put across, okay, US FDA guidances on the development. But again, when you develop a molecule, we get to interact with the US FDA and with EMA and with other agencies on a lot as a through meetings. So what we read in the guidance might not percolate in actual practice. So they might suggest you to do an extra study. They might tell to remove an extra study if they are, if they're convinced that you are in the right path. So that is how we basically design a product. It's not like, guideline driven alone. We have a lot of conversations with the agency against which we are planning to market the drug. Example of biotechnology products, there's all those peptides, enzymes, radioisotopes, all your other molecules. So product knowledge and control, the three checkpoints that we do is adequacy of the methods. Adequacy of the methods, when I say, is the entire manufacturing process, the analytical methods that we use is supposed to be very sensitive methods, to detect impurities for specific product attributes. What we have set up, it has to adequate enough. The tests capable of detecting modifications. Now, when you have manufactured product, you keep the product on stability. So right, the shelf life of a product, for example, is 24 months. 2 years we can use that. So when you manufacture a product and when you keep it on shelf life, after like 3-4 months, the product might start developing changes, right? So it might start developing fragmentation, aggregation, oxidation, all these kinds that develop that might compromise on the quality of the product. And that is the reason we keep it on stability. That is keeping on stability for 2 months, 2 years. That is how we design the shelf life of the product. So you should have methods which are capable to detect the modifications. And also, are they capable of detecting all impurities? When we say impurities, no drug is free of impurities, right? So even the novel product, any product that is in the market that have has impurities because it goes through multiple, containers, multiple manufacturing process and steps. But the impurities has to be within the permissible limit. There is no drug which has zero impurities. Impurities are allowed, but within the permissible limit. So all these are kind of studies that we give to the agency to prove that your drug is safe and efficacious. The key concepts, as I told earlier, it's a very stepwise development. Approvability is based on the totality of the evidence that you submit. Analytical similarity data is the foundation. The nature and scope of clinical studies again varies according to the product to product. So now there is something called CQAs, what we tell, critical quality attributes. So for a drug, a critical quality attribute are properties that can compromise on the quality, safety, microbiological, biological properties of the product. Now you have a product, for example, pH, or for example, temperature. If the product is supposed to be stored at 2 to 8 degrees Celsius, you have to store the product at 2 to 8 degrees Celsius. You cannot keep it at 30 degrees Celsius and expect it to behave, in the normal way. So all these are, kind of critical attribute. We have to define it right at the beginning of the stage of the development. Select, develop sensitive analytical methods, define the right QTPP. Now, QTPP is nothing but quality, target, product, profile, which we develop for biosimilars basis the novel molecules. We study the novel molecule. We understand which are the main product profiles of, for example, your pH, for example, temperature, for example, appearance, all these, qualities. We make a quality target product profile. Again, we submit it from regulatory to the R&D scientists sit over it, work over it, extract it more, refine it more. So it's like a huge process. Develop process robustness. When I'm saying process robustness, it is, as I told earlier, one batch, if you're producing, it cannot have different features from the rest of the batch. The manufacturing process has to be robust enough that whatever batches are you manufacturing, it has to be consistent in nature. Focus on achieving the quality goalpost. Quality goalpost, when we submit to FDA, US FDA gives us a timeline of 10 months for approval, okay? The approval timeline is 10 months. Within the 10 months, they will keep asking us queries. We have to address, the regulatory addresses the queries and submits to them till the point of fact that they're actually convinced, okay, that this drug is proper. So quality goalposts we expect as regulators and as company, to get an approval within 10 months, which is very rarely the case, which for biological molecules is very difficult because there are a lot of multiple queries agencies raises over the manufacturing process, over the product nature and everything. So by the time we address, my experience has been I have never got a goalpost within 10 months. It has been always dragged to like an year, at least minimum an year. Demonstrate process scalability. Suppose you're having a 20-liter fermentation, batch limit, production limit, increasing it to 40 liter, increasing it to 60 liter. That is a process scalability that you do. A thorough comparative analytical assessment. As I told earlier, the basic bridge of your dossier is your analytical assessment. So that is my last slide. I had to rush through the last slides, otherwise I will not finish before 5. Now, last few minutes for your questions. Any questions on regulatory perspective as a career or anything, please open it up.
Yeah, hi ma'am. This is Dr. Ganesh here. Just a quick question, rather curiosity. Maybe you could kindly advise. I heard you mentioning that there are likely, there won't be any animal trials with regard to biosimilar and as well as the generic medication. So in such cases, usually human subjects present with symptoms, which would be elicited in the form of signs by the physicians or the GP. Based on that, there would be a visual diagnosis followed by the final
You're talking
diagnosis.
about biosimilar molecule or you're talking about novel molecule?
Yes, biosimilar only, biosimilar case. So in those circumstances, when we...
Last part, after that, what did you say about the physician?
Yeah. See, in order to arrive at a diagnosis, usually we have a combination of symptoms and signs followed by the provisional diagnosis and then the final diagnosis.
Absolutely.
Traditional approach. When it comes to the animal trials being excluded, there would not be any physical symptoms being expressed by the patients in that context, right? So would that be a hindrance in order to arrive at the efficacy of the drug?
No. I'll tell you, when it comes to good question, when it comes to a biosimilar molecule, I'll tell you what is the entire concept of not having clinical study. It's not that I told that the animal studies can be ruled out, but FDA Modernization Act is basically speaking to rule out the animal studies even for novels and biosimilars. It's not for biosimilars alone. So now for biosimilars exclusive, the cases, the innovator has already done extensive animal studies, right? So animal studies from perspective of toxicity, from perspective of your lethal dosage levels, everything they have studied. So we basically rely on that data.
Okay.
So we don't have to repeat the animal study. So we only do the bridging clinical studies that is required to prove the compatibility of your product with the innovative product that is in the market. So now when it comes to novel molecules, I'll tell you what the FDA Modernization Act speaks about. You can Google it and also read. There are a lot of articles in that. It has not come into picture, but during the Biden's governance, they had actually put it across. So what FDA Modernization Act tells us that the animal studies, because we have by now over years of consortium of data we have on animal studies, right? So whenever it's not like they're going to exclude it, but very minimal animal studies to be done for novel molecules also. So because we have a lot of these in silico modeling, we have a lot of analytical statistical packages that can actually mimic the entire pharmacokinetic and pharmacodynamic behavior within an animal on the data for a typical indication, for a typical disease. So basis that, they are working out ways on how they can reduce the animal burden from that aspect. So answer your question?
Yes, that's quite informative. Thank you very much. Now
you can read that. FDA Modernization Act 3.0. You can just Google it.
And regarding the second part of my question related to the regulatory affairs, see, for instance, if a drug is being introduced after all the thorough prerequisites and all in one country, and the same drug, after getting all the required permissions and authorizations, is being launched into a different country or 2 or 3 other countries. So if there are no side effects or any untoward results being reported in the native country, but some of the issues arise in the form of side effects in the other countries where the drug also has been later launched. So where should legally it should be filed or reached out to? Whether
a very typical physician question, no? I'll tell you why. So now, suppose in India, I am launching a biosimilar. I have to do it on an Indian population. You get that part, right? So I have to do it at an Indian population. My bridging clinical studies, I cannot do it on a Chinese population and launch my product here. Okay, agreed on that part? Now I'm launching my product here. The same product, I am trying to percolate it in China now. I have to do a Chinese population study. I cannot reflect the studies over here to the local population. So now I'm supposed to be doing it in Russia. I'll have to do it in a Caucasian population or whatever race of population there are. So that without doing the region-specific clinical studies, we do not generally get approval for at least the bridging minimal clinical studies. We will have to do it in a local population. That is when we launch the product there, once we get the approval.
Excellent. Thank you very much. That really helps.
Anyone else? Any questions? We are done with the time, but I can sit for another 5 minutes also if Shweta allows.
All yours, ma'am, please.
Hi, Sweety Mathew. This is Amal. We did Global Regulatory Affairs.
Oh, yeah. Hi.
Hi, how are things?
Good, good. Good to see you.
Yeah, I have a question regarding precision medicine in India. Like, how is the current landscape? Are we waiting for
medicines, did you say?
Precision medicines.
Precision medicines, okay.
Go ahead.
No, I understood. What was your question?
So what is the current landscape of precision medicine in India?
See, personalized medicine we have a lot of evolving regulatory for personalized medicines, but there is, a regulatory requirement in India as such. It's evolving. There is a lot of gap even in Indian regulatory agency even now. When you're speaking about specific gene therapies, specific stem cell therapies, this is a lot of focus area, but we have a lot of gap in India in the regulatory guidances perspective. And similar ways in other countries also, nobody's like, it's not evolved, but EMA and other agencies, FDA has a lot of papers on precision medicines, but in India, there is a lot of gap now. I'm currently planning to pursue my PhD in precision medicine, so I'll let you know later when I have something solid in picture. So as of now, there is a lot of gap.
I am currently working on a pricing strategy of precision medicines. I mentioned to you before.
Right.
Yeah, I'm almost winding up the thesis at the moment.
Okay, great. Thanks, Amal.
Thank you. Thank you.
Anyone else? Any questions?
Keep asking us about regulatory affairs and the career prospects when you have
it does not have to be around technically surrounded, you can also ask on your career
Yeah.
perspectives and current corporate scenarios for regulatory affairs, scopes, everything.
Ma'am, Kankana here.
Yes.
So actually, what's the required skill set to get into this field, this regulatory affairs?
Okay, so I'll tell you my example quickly. Okay, so I was part of R&D. Getting into regulatory was very difficult for me because I did not have the experience. But it happened very fortunately for me that they started a new wing for peptides. So now in Biocon, that is how I joined them. But the thing is, there are a lot of regulatory specific courses which Biocon is running. There are a lot of companies which run. So some sort of regulatory understanding is required for you to get into the wing, which is fair enough, right? I mean, a person who does not have any regulatory experience to get into regulatory, or even to the matter of fact of freshers, the freshers, there is a very good scope because there are a lot of companies giving you internships. So get into the internships, and you can always explore into any vacancies at that time. It's a little challenging, and it's not in regulatory, it's in any field, right? To get into as a fresher, to get into the the industry, and at the same time switching off departments. So I always say the one department which always I used to feel that the entire drug cycle in a manufacturing scenario was the marketing, was the manufacturing, sorry, the productions. In productions, people who are working, there are a lot of people I know who have shifted from productions, who have shifted from R&D and made a switch to regulatory. But it's also regulatory's key specialities or key lookout features are very strong communication skills, written skills, being very confident, because there is a lot of what do we say, interactions that we do with the customers, interactions that we do with the regulatory authorities. So that is the major fetch out. Regulatory science is not a rocket science. Anybody can learn it, but getting into the right fit is very important. So there are certain certain what do you call that, priorities that they look out for are these features: very strong communication skills, able to interpret data, able to understand guidances, very sound understanding about things. So these are generally getting an interview. You will get an interview basis, you always have an understanding of whatever drug, whatever pharmaceutical vertical you are in, you can always make a switch. But for freshers, again, internship is one of the options. Any of the courses that they do, it adds to the benefit.
Hello, ma'am. Nikita this side. Ma'am, mostly when I searching for a job in the regulatory affairs, mostly their requirement is pharma graduates, and I am from the life science post graduation in life science. So it is quite hard to find a job in a regulatory affair or not as
I am not a
compared to.
pharma graduate, Nikita. I am a life
Okay.
sciences graduate. So there is nothing
Okay.
like pharma graduate, but generally people, especially when you're active pharmaceutical ingredient regulatory team, they would prefer people from medicinal chemistry having a pharma background, because M-Pharms and all, they have better understanding about the medicinal chemistry, right? So there are certain people who keep requirements like that, but there is no requirement as such that. As long as you prove you are smart enough to fit in the team, I don't think there is anything that can be challenging.
Okay.
As a fresher,
Understood.
are you a fresher?
Yes,
So
I
as a
have
fresher, I
experience
told
in
earlier,
medical side.
as I told you earlier, as a fresher, it's quite challenging, not only in regulatory, in any field in pharma, right? So
Yes,
there are a lot of internship opportunities that you get in Biocon, and a lot of other companies that they provide. Try to get into an internship opportunity and understand the dynamics of the workforce. Then it's easy for you to apply for vacancies, and then you have some reliable, short experience in your CV to show.
yes, ma'am. Thank you so
Yeah.
much. Yes, ma'am.
Right, I think that's about it. It's the last question. Anyone else? I'm very active on LinkedIn. Please reach out to me on LinkedIn if you had any questions regarding any of the context that I have taken in the slides. Be happy to reply.
Hello, ma'am. I think that would be about it. And before you leave, any message for these young budding healthcare professionals? I mean, that would be a great use for them. And also, thank you so much. It was, I mean, we were absolutely honored and thrilled to host you. The session was very good and very detailed. Thank you so much. Message before you sign off would be great.
One thing I'll tell you what I follow: being in a field, you have to enjoy what you do. Okay, knowledge increase is very important. but knowledge sharing is very important. Anything that you learn, keep reading, because it's very difficult if you open a US FDA guidance and read. Even today, I cannot read through a guidance and sit and understand it. That's not happening, because it takes years for them to frame a guidance. So it's very unrealistic to expect someone to make you understand a guidance in a day or two, right? So it is not that. It is like, be aware of, for example, I told you about this FDA Modernization Act, right? A lot of people won't be aware of it. So keep your eye open. There are so many things to read and, to understand, have an insight, learn and share. That is when you share, that is when you increase your knowledge. So getting into regulatory as a career, it's a brilliant platform to be in. And any guidances you need, please reach out to me on LinkedIn. I'm very active on LinkedIn. I can respond, probably taking a few days here and there. I had a good time sharing the info. I don't know how much did it percolate through the information. I hope people had at least a quick overview of what regulatory was and what do we do as part of the industry. Thank you so much for hosting
Thank you
us.
so much, ma'am.
Thank you. Have a
Thank
good
you, ma'am.
evening, all of you guys. Thank you. Bye-bye.